All right, welcome back, everyone. Our next speaker is Huimin Zhao, who was the Stephen L. Miller Chair of Chemical Engineering at the University of Illinois at University of Illinois Urbana-Champaign. He received his PhD in chemistry from Caltech with Nobel laureate Francis Arnold, and he worked at Dow Chemical as a project leader in industrial biotechnology and has been on the Illinois faculty since 2000. So much to say, I got to turn the page. Professor Zhao is a pioneer in directed evolution, enzyme engineering, and synthetic biology. More recently, he has led the integration of artificial intelligence, machine learning, and laboratory automation into biocatalysis. His group has developed breakthrough methods such as photoenzymatic radical hydroalkylation, AI-enabled retrobiosynthesis, and contrastive learning for enzyme function prediction, work that has been published in Nature and Science. Today, he will speak on expanding the boundary of biocatalysis from directed evolution to AI-enabled autonomous experimentation, highlighting how the convergence of enzymology, photocatalysis, and AI is opening entirely new frontiers for sustainable synthesis. Welcome.
Okay, thanks for your nice introduction. And it's my great pleasure to be here again. I visited this campus actually several times and also have many actually friends on campus. So today actually I'm going to talk about our recent work in the synthetic biology area with the focus on enzyme engineering. So my group is really work at the interface of synthetic biology, AI, machine learning, and the laboratory automation. So we're very interested in developing like a foundational tools for synthetic biology applications. Particularly actually is the workflows, right, for enzyme engineering and metabolic engineering, and also actually AI tools or machine learning tools that we recently developed for enzyme engineering. And also we try to integrate those tools actually in the form of a biofoundry, which I will talk about in a minute. And then we try to leverage this technology platform mainly for three main applications. One is actually related to drug discovery and development. We try to identify bioactive compounds from soil bacteria and then study their mode of action and also biosynthetic mechanisms. And in the second major application area is related to microbial cell factories. So we try to engineer microorganisms for production of chemicals and materials. And then lastly, we're also interested in mammalian synthetic biology, in which actually we developed many genome editing tools for the study of chromatin structure and dynamics and also for gene therapy applications. But today actually I'm going to talk about two actually stories. One is related to enzyme engineering. It's called, we try to develop enzymes with new to nature reactivity. Particularly, we try to combine photocatalysis with enzyme catalysis to create artificial photoenzymes that can catalyze reactions that cannot be catalyzed by natural occurring enzymes for sure, but also very hard or not possible by chemical catalyst. And then the second story is related to AI-driven autonomous experimentation for enzyme engineering or for synthetic biology application in general. So in this paradigm, we actually try to use AI tools like large language models or generative AI tools to create library variants, followed by the experimentation by robotic systems. We call it biofoundry. And then we use AI tools to make predictions, and then we can repeat this cycle again and again, right? So it's AI-driven autonomous experimentation paradigm. So we wonder why we're interested in enzyme catalysis. Probably I don't need to explain the reasons, because for this audience, we are interested in enzyme catalysis, right? Because compared to chemical catalysts, enzymes have quite a few advantages, but the biggest advantage is really the selectivity, right? Because this is very hard to do, by chemical catalysis, but also, they are environmentally friendly and easier to engineer. But for chemical catalysis, their biggest advantage is really the broader reactivity. They can catalyze many different reactions, right? So there are actually two kind of grand challenges in biocatalysis. One is that how we can efficiently create enzymes with new or improved functions and particular reactivity, because that is the biggest limitation for enzymes. In many cases, we just couldn't find the enzyme that catalyzes the reaction we want. And then the second major challenge is that we know each type of catalysis has advantages and disadvantages. Then the question is how can we combine the advantages of two types of catalysis? So we started this effort actually more than 10 years ago, even for the enzyme engineering, started when I was a graduate student. But for the second challenge, we actually tried to combine biocatalysis with chemical catalysis by taking the advantages of both types of catalysis. And we found that it's very challenging, actually. I think it's easily said than being done. I think the biggest challenge is really the incompatibility of two types of catalysts. As we all know, enzymes work in aqueous solution while chemical catalysts work in organic solvent, high temperature and pressure. So it's hard to put them together. So initially, we came up with a strategy to actually combine olefin metathesis with enzymes to synthesize some enantiomer selective compounds. And then later, we found that actually photocatalysis is kind of inherently compatible with the enzymes, right? We demonstrate that we could actually include a photocatalyst with an enzyme in the same reaction mixture to achieve a symmetric synthesis. And inspired by that, but also my former lab mate Todd Heist also is a pioneer in this area as well, try to actually really develop enzymes with new capabilities. So the idea actually is very simple in retrospect, right? So we know that photocatalysis is very versatile. They can actually react with all kinds of, because it will generate the radicals, free radicals, and the radical can react with many different functional groups. While for enzymes, it has the active site with a chiral environment, so it's very selective, but it's not very reactive. That's why we try to combine these two types of catalysis in a single enzyme to create a new class of artificial photoenzymes. The way we do it is really just use the light to create the radical on the cofactor in the enzyme, like flavin or NAD, right? And then we use the active site environment to control the selectivity. So this strategy actually works very well. We demonstrate as early as 2020, we could actually engineer an enzyme to catalyze intermolecular radical hydroalkylation reaction to synthesize chiral compounds, which actually is very hard to do chemically and clearly cannot be done by enzymes. And then later we also showed that we can also achieve intermolecular radical conjugate addition reaction. More recently, we showed that we can achieve the asymmetric incorporation of fluorinated motifs to the target product, which I will talk about in a minute. In addition to these carbon-carbon coupling reactions, we also showed that we could achieve carbon-nitrogen coupling reaction, meaning the hydroamination reactions as well. And also we can achieve high selectivity. And all these reactions actually follow the same biosynthetic mechanism, Essentially, we use the light to create a radical on the cofactor, as I mentioned before, but also create a cofactor in one of the substrates. So we have a radical substrate, and then we have the olefin substrate. And then these two substrates can react with each other to form prochiral compound. And then the enzyme really control this hydrogen atom transfer step to actually achieve the selectivity in the target position. Right? So that's actually the mechanism. And I can show you actually the first example is the fluorination reaction. We actually didn't do direct fluorination. We just incorporate the fluorinated motif, to the product. And as you know, and of course Michelle is expert in fluorinase. In nature, there's only one class of enzyme called fluorinase that can catalyze this reaction, and it's very rare. But we know that the fluorine has a unique properties. Once you incorporate it into the product, it can change the physical property, make it the product more bioavailable. And that's why in a lot of pharmaceutical or agricultural chemicals, right, they all contain the fluorine in it. And the strategy we use is basically use a substrate that contains the fluorine, also the leaving group. And then we use the light to create the radical containing the radical substrate containing the fluorine. And then we do the carbon-carbon addition reaction, and then we can introduce the fluorine to the product at different positions. And initially, we actually screened a number of those fluorinating agents because we don't know which one work, and we found this trifluoroiodine ethane, actually that the TFIE worked, right? It showed, at least the product. The yield is very low, but the EE is pretty good. And then we also made sure that this reaction is truly enzymatic reaction, right? We actually did all the control experiment, and it is clearly the enzymatic reaction. And then we also tried many different substrates to show the broader specificity. And most of compounds are still related, but nonetheless, we could show that the compounds can be all achieved with the high EE, because that's very important, and also a very good yield as well. Some could be as high as 94%. Some could be like a 34%, but it varies. And we also actually did some further engineering as well, mainly for the ones that the selectivity at the more remote site, like the gamma position, right? The beta position, I think we can do it, but the gamma position or the delta position for those remote sites, the natural occurring enzymes just couldn't catalyze those reactions very well. So we have to perform some direct evolution studies to improve the yield and selectivity. But in the end, we actually could obtain those engineered enzymes that can catalyze all these asymmetric reactions. And then we also did the computational studies as well, really tried to figure out the mechanisms, right? We found out that these two particular residues, the tryptophan 116 and the threonine 37, are involved in the control of the sterol selectivity. And then we also explained the selectivity of the reaction as well. Okay, now would like to switch gear. I will talk about the AI-driven autonomous experimentation platform for enzyme engineering, or in general, synthetic biology. So this paradigm actually is very similar to direct evolution. So in direct evolution, we basically use a random homogeneous or recombination to create a library of variants, followed by high throughput screening or selection, and then we find the best one, then use that one as the starting point for the next round, and we do it iteratively. While in this autonomous experimentation paradigm, we use large language models or other AI tools to generate a library of variants, and then use the biofoundry or robotic system to carry out all the experiments. And then we use the supervised machine learning model to make predictions. So we don't throw out all the data. We will take all the mutants' data into consideration to make new predictions, right? And then we can do it iteratively. And this new paradigm, of course, is my favorite, because my PhD thesis, as Ramas just pointed out, really focused on direct evolution, right? So I developed a number of technologies for direct evolution, now have been widely used. But the concept of direct evolution actually is kind of similar to the concept of autonomous experimentation. And we started this effort more than 10 years ago. At that time, I was inspired by the development of computer on our campus. So more than 70 years ago, a group of faculty from our university designed one of the first computers called ILANAC, or Illinois Automatic Computer, which actually consists almost 3,000 vacuum tubes, weighed about 5 tons, right? But yet the computing power of that computer already exceeded all the computers combined in the Bell Labs at that time. So then we actually designed a robotic system called Illinois Biological Foundry for Advanced Biomanufacturing, or iBioFab in short, for rapid prototyping of biological systems. And this effort actually led to the formation of a Global Biofoundry Alliance in 2019, which currently consists of more than 40 members around the world. We just had the annual meeting last week, just a few days ago, at Illinois. And then also this effort led to the formation of NSF Biofoundry, iBioFoundry, or Illinois Biofoundry last year. So the system we developed essentially is a fully integrated robotic system, and you can think it is an automated lab. And our idea is to basically we integrate more than like 20 instruments into a larger platform. And as shown in the video, in the center of the platform, there's a robotic arm that takes a 96-well plate from one instrument to another. And those instruments are mainly used for synthetic biology kind of experiments. So idea is to break the workflows into like protein engineering, metabolic engineering into process modules, which will be in turn broken down into unit operation, process modules, which will be in turn broken down into unit operations. And then we'll program those unit operations to create a custom designed workflows for different applications. So we started this effort more than 10 years ago with the funding from Carver Trust. It's a non-for-profit organization. And then in the past 10 years, I raised the funding from various funding agencies, including DARPA, NIH, DOE, and SF, to support the development of the capabilities of the BioFoundry, but also explore the applications of BioFoundry in various areas. And here I just show one example in which actually we demonstrated this AI-driven autonomous experimentation paradigm. It is a very simple proof of concept. We use a pathway that is involved in the synthesis of lycopene that consists of only 3 genes. And then we use promoters with different strengths, try to optimize the flux. So in total, there will be more than 13,000 combinations. We could use the robotic system to construct all these variants, but it will be time consuming and also expensive, right? And then we decided to incorporate the AI basically to shorten the experimental effort, to reduce the experimental effort. And what we did is that we constructed a very small library, actually only like 46 variants, and then use the robotic system. And then we also characterize the mutants using the robotic system, the mutant pathways using the robotic system. Then we use the Bayesian optimization to predict mutation, to predict the combinations of promoters that can lead to high production. And this effort actually led me into the AI field about 8 or 9 years ago, right? So we didn't jump into the AI field because now it becomes so hot. We jumped into the AI field because the need to really demonstrate the power of the BioFoundry. And so in the first round, we actually selected, made only like 46 pathway variants, right? And then made the plasmids and then do the transformation and the characterization. So all these steps were done by the robotic system, right, with no human intervention. That's why I said it's the autonomous experimentation, right? All the experiments can be done by the robotic system. And we repeated this cycle actually 3 times, and we found some mutants showed greatly improved production. Right? And we also did a control experiment just by random immunogenesis. We could also create a library as well, but we found that our method is much more efficient because we only evaluate less than 1% of all the possible combinations to find the best combination. And this work actually led to the formation of the NSF AI Institute more than 5 years ago, right? That actually was fortunate. I was pretty fortunate to actually lead this AI Institute, which is one of the first 7 AI institutes funded by NSF. Currently, there are 28 AI institutes funded by NSF, covering different areas. But this is still the only one that is focused on chemistry. So basically, in this institute, we try to develop a new AI and new chemistry or biochemistry to accelerate and democratize molecular synthesis and also functional material discovery. So we have 5 thrusts. In the first 2 thrusts, we focused on the development of AI tools for synthesis planning and catalyst selection and optimization. And we're particularly interested in combining chemical catalysis with enzyme catalysis, right? Try to come up those most efficient synthetic route for the synthesis of targeted small molecules. And in thrust 3, we focus on 3 molecules. 2 of them are drugs. The third one is a molecule used in polymer processing, and we try to come up a synthetic route for the synthesis of those compounds. And in thrust 4, we focus on organophotovoltaic materials, and we try to improve the stability of those materials. And we also established an industrial partnership program currently consisting of several pharmaceutical companies, IT companies, and biotech companies. And half of the PIs in the institute are CS faculty, half of them are chemistry and chemical engineering faculty. And luckily, we got it renewed just a few months ago for another 5 years. So we also have many collaborators in the country as well, and also have a very strong external advisory board that consists of experts from industry, academia, and non-for-profit organizations. And also we trained many graduate students and postdocs in the past 5 years. And one key deliverable in the institute is the alpha synthesis platform. This is an umbrella term, right, for all the AI tools we develop. So basically, we try to develop AI tools that can be used to mine literature to gather important information about the chemical reactions and also enzymatic reactions or the mutants, the protein mutants. And then we also leverage the existing database as well as the data that we will generate using our BioFoundry or similar system, robotic system for chemical synthesis. So we then integrate all of this data, right, to create a database that will be used for the development of AI machine learning tools for synthesis planning, catalyst selection, and optimization. So in addition to this platform, in addition to this alpha synthesis, we also developed a new paradigm called AI-powered closed loop. It's similar to the AI-driven autonomous experimentation, but for chemical synthesis, sometimes it's harder to achieve autonomous, right, experimentation. In many cases, you still have to do it separately, but for biological experience, like enzyme engineering, clearly it's possible. So basically now we can use AI to design and guide the experience, and then use robotics to perform all the experience. So here, just highlight a few AI tools that we developed. One actually is mentioned by Romas already. So it's a deep learning tool that can be used to predict the EC number, the enzyme commission number, that will indicate the reactivity of the enzyme and also the substrate, right? Using this tool, now we can predict the enzyme EC numbers for those with very low homology with the known enzymes. Because currently, most people just use the BLAST P to predict the protein functions, right, based on sequence analysis. And this, and also many of you know, the AlphaFold now is widely used for predicting protein structure from sequence, but that cannot be used to predict function. Right? And so in this new tool, we actually use the contrastive learning algorithm. Basically, we use the enzymes in the Brenda database as the anchor sequences, and then use all the 500,000 characterized enzymes as the testing data sets to train the machine learning model. And our goal is to minimize the distance between the positive and the anchor sequence and maximize the distance between the negative and anchor sequence. So we trained the model with many of those enzymes. And then when you have an unknown protein sequence, we actually can calculate the distance, basically, and then assign an EC number, right? And we actually did benchmark studies with all the other known, existing machine learning models, and also bioinformatics tools like BLAST P. And we found that our tool outperformed all of them. And particularly, we also did experimental validation as well. We used a heterogenesis as a case study. We have more than 400 those heterogeneity that have not been very well characterized. And we found that some of them were misannotated. They basically thought that they could have only one reaction, or either only catalyze a chlorination reaction instead of fluorination reaction. So we actually synthesized those genes and then did the enzymatic assay to prove those gene annotations was incorrect. And then we also actually benchmarked the other tools. Sorry. And we made this tool actually publicly available. The users just need to enter a protein sequence, and then you can get the predictions very quickly. So I think users all over the world are now actually using this tool. But then more recently, we actually just have another AI tool that actually will be published very soon. And in this tool, now we can predict the enzyme subspecificity. So in many cases, you already know the enzyme can catalyze that reaction, but you also have many substrates, right? Or many small molecules. You don't know which one will work well, right? So then we actually developed this deep learning model that leverages the protein large language model and also the chemical large language model. But particularly the innovation is the cross attention layer that really look at the interactions between the protein and substrate. And then we also did many docking studies to create a data set, because the existing data in the PDB data bank for protein small molecule interactions are not sufficient for machine learning model development. So we did a very comprehensive studies on that. And once you have the enzyme that works for a certain reaction, but in reality, in many cases, they still don't work very well, right? So we also, a few years ago, developed a deep learning model for enzyme engineering. So in this model, we actually look at all the protein sequences in the UniProp database, and also look at the homologous sequences for the target protein, and also look at the mutants. So basically, we combined the data set, 3 data sets, into a single deep learning architecture to predict the mutations that can affect protein functions. And we also did the benchmark study, experimental validation as well. But the field develops so quickly. Almost every month you will see another new machine learning model for protein engineering. So it's hard to catch up. But just a few months ago, we also demonstrated that we could incorporate or integrate AI with robotics and synthetic biology to achieve autonomous experimentation for enzyme engineering. So in this case, we use a protein large language model to make predictions on the mutation that can affect the protein function, and then use the robotics system, the biofoundry, to carry out all the direct mutagenesis study, right, to create the mutants. And then we also characterize those mutants using robotics, and then make predictions using supervised machine learning model. And then we do it iteratively. But also we developed an AI agent by leveraging the ChatGPT, so then researchers can also talk to that agent to help you to design the experience as well. So in this platform, we actually only create a very small library of variants in the first round. Here we use 2 enzymes as test case. One is phytase, the other one is methyltransfer. So we only create like 180 mutants that are predicted by the protein large language model for improved protein function. And then after we screened the library, we found that many of them show the improvement. And then in the subsequent round, we use supervised learning model to predict the combination of those mutations that can lead to further improvement. And then we only made less than 96, or about 96 variants in each round. So in total, we only made less than 500 variants, yet we can obtain the mutants show the significantly improved activity for both enzymes, right? And also we found that the machine learning model did a much better job than a human intuition. I will skip that because this work is already published. And we did a detailed characterization as well. But this, of course, we continue to develop new capabilities for the biofoundry. In the DOE center that I'm involved, we try to develop new AI and machine learning tools, and also the self-driving biofoundry for enzyme engineering and metabolic engineering. So we have a team of PIs from 7 institutions. And also, as I mentioned in the beginning, we launched the NSF Illinois Biofoundry just a few months ago. So our goal is to develop a cloud biofoundry that is seamlessly integrated synthetic biology, AI, and automation to democratize basic and applied biological research. So our ultimate goal is to make a user facility, right? So we'll work with users from academia and industry, try to encourage them to use our technologies, of particularly the facility, but also we will work with them to develop new technologies as well. And then we also have the internal research program focusing on developing new tools. We just had the ribbon cutting event like a few days ago at Illinois. So this is brand new, but I definitely encourage you to contact us, work with us. We are very eager to work with anyone in the US. And then our ultimate goal is to really develop this AI, machine learning and automation-enabled pipeline for the synthetic biology. There are many steps in this, almost all the steps in this synthetic biology pipeline could be enabled by AI machine learning tools, and also many of the steps could be facilitated by the robotic system. So I see there's a lot of opportunities for this direction. And also last year, I launched another NF center called the Global Center for Biofoundry Applications. So we also work with the biofoundries from other countries, including UK, Japan, Korea, and Finland, right? We try to work together to develop the standards and matrix for the workflows to make our biofoundries basically more reliable and scalable. And this is also very new. But my ultimate goal, as you can imagine, is really to establish this AI-driven autonomous experimentation pipeline in which we can actually use AI to decide what to make, how to make it, and also figure out what that means. Particularly, it can also be used to generate hypotheses. So actually, a scientist from Japan even predicted that maybe in the future, AI itself could win a Nobel Prize. And of course, this is a very new field. So we actually really try to promote the development of this field. So as a chief editor for the SS Synthetic Biology, we have a recurring special issue on AI for synthetic biology. And also a few years ago, I organized a workshop for DOE to really discuss the opportunities and the challenges in AI and machine learning for synthetic biology applications. So the report is publicly available. You can read it. So in summary, what I talk about today is that we have been developing photo biocatalysis coupled with direct evolution as a promising strategy to create enzymes with new to nature reactivities. We had quite a few case studies, but also we established an AI institute focusing on development of AI tools for molecular discovery and synthesis, and showed a few AI tools that we developed. Today, I only talk about the AI tool for predicting enzyme function and also the specificity, but there are many other tools as well. But the take-home message, or the most important message I try to convey, is really this AI-driven autonomous experimentation paradigm. I think this is a very powerful paradigm for many researchers. And finally, I would like to thank the students and postdocs who did most of the work, and particularly funding from DOE, which provides most of funding for us to develop the capabilities for the biofoundry, and also NSF, for the development of all the AI tools I just talked about today. And thank you for your attention. I'll be happy to answer questions. Yeah.
Thank you very much, Huimin. We have time for one question.
Okay. Yeah.
So the success of AlphaFold is not just the amount of data that's available, but also very well curated, consistent data available in PDB. For catalysis, enzyme information is somewhat inconsistent and not very well curated. So what do you think is the major challenge in making AlphaFold or alpha synthesis that you already made? And you also kind of showed your example for lycopene synthesis, but even using robotic system, there's about like 46 plasmids that you generate in an iterative cycle. Can you actually increase that number to like hundreds or thousands or even like millions of variants at a time to really generate that well-curated, consistent, reliable data so that people can benefit from that?
No, I agree. That is a major limitation. The reason AlphaFold became so successful, it is because built on the more than 40 years research, right, on the curated in the PDB data bank. While for enzyme functions, indeed, we don't have this kind of database. So recently, we launched another effort to really try to establish called an open enzyme database. We try to work with the broader research community, ask them to deposit those high-quality data. But also, we need to rely on those biofoundries, to characterize those enzymes. And this is something already being done by NIST and Align, non-for-profit organization. They kind of started some effort. But I think this probably requires a community-wide effort. And currently, we just curate all the data from different databases. But I think it's important to generate new data as well. Because the more data, particularly the high-quality data, is critical for the machine learning models. Yeah.
Please join me in thanking
Yeah.
Huimin
Okay.
Zhao.
Thanks.